Now, the cell membrane is semi-permeable, which we also call selectively permeable. And the word permeate or permeable means able to pass through or able to cross. So selectively or semi means that some stuff can cross the cell membrane, but not everything. So some stuff can make it across, but not others. So for example, if we've got a couple of cells here, there are some things that can enter and leave all cells, and we'll talk about what those types of things are on the future slides. There are other things that can't get into the cell, or if they're inside the cell, they might not be able to leave the cell. But the interesting thing is that this permeability varies by cell type. So some cells are more permeable to more substances than other cells. So perhaps in this example here, maybe the blue and red stuff is not able to cross this cell. Whereas in this cell, maybe whatever the blue material is not able to cross, but maybe the red stuff is. And then maybe this cell down here has a lot of permeability where both the red and the blue and the green can cross the cell. So it kind of depends on the types of cells. And some examples of differences might include things like if you think about the skin. So the skin is meant to be a barrier and it's not very permeable. It does have some permeability because there's some stuff that can enter and leave all cells. So it does have some of that permeability like what we see with the whatever this green arrow is representing here. But for the most part, your skin is trying to be a barrier and is trying to keep stuff inside your body and out. It's trying to keep stuff inside your body that's supposed to be in your body and stuff outside your body that's supposed to be out. But by contrast, you might have an intestinal cell. And intestinal cells have a lot of permeability because this is where you absorb nutrients into your body. So you need to be able to absorb things like glucose and amino acids and proteins and fats. And so there needs to be more of that permeability. So on the one kind of side of the spectrum, you've got the skin, which has very little permeability. On the other side, you've got the intestines, which have a lot of permeability. We mentioned in the previous slide that different cells have different permeability. And so let's explain how that happens. So first of all cells have a phospholipid bilayer. So remember, phospholipid bilayer is the majority of the cell membrane is made up of these phospholipids. So if we were to, I'm going to draw a little area in here. So if we were to draw this phospholipid bilayer, we would see the phosphate heads on the inside of the cell and the outside of the cell membrane. And then we have these lipid tails that are in the middle. So remember, this would go all the way around. I'm just being lazy by drawing just a line for the rest of the cell membrane. But this phospholipid bilayer is the same regardless of the type of cell. So whether it's an intestinal cell or a skin cell, all cells have this phospholipid bilayer. So certain things that can cross the phospholipid bilayer are going to be able to cross those phospholipids regardless of which type of cell it is. All right, so whether we're talking about a skin cell versus an intestinal cell, like we mentioned on the previous slide, the phospholipid bilayer is the same in all cells, and it means that that same permeability. So if something can cross the phospholipid bilayer of one cell, it's going to be able to cross the phospholipid bilayer of all cells. Now, the difference comes into play with these transport proteins. So with these transport proteins, these kind of act as kind of like pores or doorways for specific molecules to be able to cross the cell membrane. And so these are molecules that can't cross the phospholipid bilayer on their own. But let's say, for example, that with the intestines over here, let's say that we now have a transport protein. And remember that these transport proteins are integral proteins. So they're just a more specific type of integral protein. And so they're touching both the intracellular fluid and the extracellular fluid, so the inside of the cell and the outside of the cell. And so now this molecule up here that couldn't fit across the phospholipid bilayer, instead it can come over here and go through this specific transport protein in order to get into the cell. Now the skin cell, if it doesn't have that transport protein, then that molecule is still not going to be able to get across the cell membrane. And so this is where we really get the difference between the permeability in different cells. It has to do with which proteins are present. And something to point out about these proteins is that they're very specific. And what I mean by that is that there are specific proteins that transfer specific molecules or substances. So for example, let's say that this red little dot here represents sodium. So sodium is an ion. It can't cross the phospholipid bilayer on its own. But this is a specific sodium transport protein that allows sodium to enter this intestinal cell here. Now let's say a different substance needs to be able to get across the cell membrane. For example, let's say we have a potassium ion over here. Now potassium cannot fit through the sodium transport protein because it's the wrong shape and size, and so it won't fit. Instead, potassium has to have its own transport protein specific to it. And as long as it has its own specific transport protein, it can then cross the cell membrane. But once again, it has to do with the specificity in that potassium needs its own transport protein, whereas sodium needs its own specific transport protein as well. So here's a list of some of the common things that can cross the phospholipid bilayer. So remember that these are things that do not need a transport protein. So this is going to be the same for all cells. All right, so all cells have a phospholipid bilayer, so all cells are going to be permeable to these substances. So first of all, things like oxygen gas and carbon dioxide, these are small molecules and they're gases. Oxygen is only made up of 2 atoms. Carbon dioxide is made up of 1 carbon and 2 oxygens, so 3 atoms. Water can also cross the cell membrane on its own, but it does so fairly slowly. And so we'll actually talk a little bit about how even though water can cross on its own, there are some transport proteins that help to speed up the process. So we'll talk about that more when we talk about a process called osmosis. Other things that can cross that phospholipid bilayer are going to be things that are hydrophobic, nonpolar, and lipid soluble. And these 3 terms go together because things that are lipid soluble are hydrophobic, things that are nonpolar are lipid soluble, etc., etc. And so we're primarily talking about things that are lipids or fats and things that can dissolve in fats. So things like steroid hormones are types of fats. There are some vitamins that are fat soluble that can also cross that phospholipid bilayer. So remember, these are the substances that are crossing that phospholipid bilayer on its own without any help. Now remember that the thing that does vary by cell has to do with the presence or absence of the different types of transport proteins. And so examples of things that cannot cross the cell membrane on their own, or I guess I should say they can't cross the phospholipid bilayer on their own. So these are things that can't get across there. Are things that are going to be like large stuff. So things like glucose. So glucose is made out of 12 hydrogen atoms, 6 oxygen atoms, and 6 carbon atoms. So that's 24 atoms. So that's a pretty big molecule. Also things like a lot of amino acids are kind of big. So just in general, big stuff needs some help getting across the cell membrane. Also, things with charges, all right? So ions are these charged atoms, as you can see here with the little plus sizes, or little plus signs, I'm sorry. You can also see things like chloride, which has this negative sign. You can also look at polar things. So polar things have charges. And so charged particles and charged molecules are not able to get across that phospholipid bilayer. And so once again, things that go hand in hand with polar are things that are hydrophilic, polar, and water soluble. And so kind of, once again, sugars, which includes glucose, and things like our amino acids, they need some help. And that's where, the transport proteins come into play. And remember that there is specificity. So these transport proteins are specific as to what they can transport. So something that can transport glucose is not also going to be able to transport sodium. Sodium needs its own transport protein in order to get across the cell membrane. So remember that these transport proteins are they're integral proteins, and we can also call those transmembrane. And trans means that it crosses, so it can go all the way across the membrane. So that means that it is these proteins here are touching both the extracellular fluid and the intracellular fluid. And you can see here that they act as these kind of pores or doorways in order to allow molecules to go into and out of the cell. And as we mentioned, different cells can have different transport proteins, and these transport proteins are specific as to what they can transport. These are the different types of membrane transport that we're going to be talking about individually on the following slides. And so these are essentially different ways that basically stuff can get across that cell membrane. Now, we can divide these types of transport into two main categories, and those categories are passive and active. And the main difference between passive and active transport is whether ATP is required or not. And remember that ATP is the energy molecule of the cell. So in passive transport, energy does not have to go into transporting something across the cell membrane. But in active transport, that ATP is needed in order to transport something. So the three types of passive transport that we're going to talk about are simple diffusion, facilitated diffusion, and osmosis. And the three types of active transport are going to be primary active transport, secondary active transport, and vesicular transport, which includes things like endocytosis and exocytosis. Before we talk about those different types of transport, we first have to do a little bit of a review as to what a solution is. So a solution is where something is mixed into something else. And so this involves dissolving something in something else. And the components of these solutions are going to be solutes and solvents. And essentially, the solute is the thing that is going to be dissolved inside the solvent. So the solvent is usually water or some sort of liquid, and the solute is usually something that is solid, although there are exceptions to this because you can also dissolve oxygen in water. But for the sake of this discussion, we're going to consider the solvent to be primarily water, and the solute is going to be something that is dissolved in water. Now, when we talk about solutions, we have to talk about the concentration of the solution. And the concentration is just a measurement of how much of that solute is dissolved in the solvent. So we can kind of write this in this way. So the concentration of the solution is equal to the amount of the solute divided by the amount of the solvent. So the example I have down here is if we look at a, let's say we can make this like salt water here. So if salt water has a concentration of 9 grams per liter, that means that our salt water has 9 grams of salt, which is our solute, for each liter of water. Now we might not have a full liter of water, but this is a ratio or a proportion, all right? So this concentration is a ratio or a proportion of how much solute there is given the amount of solvent present. When the concentrations of two solutions are equal or the same, we call that equilibrium. So that means that whatever the concentration is in this area is going to be the same as the concentration in this area. So for example, if we were looking at our saltwater example that we used on the previous slide, we could say that this area has 9 grams of salt per liter of water. If there is equilibrium, it means that this area also has 9 grams of salt per liter of water. And so this is equilibrium in which these two values are going to be the same. In contrast to equilibrium, sometimes a concentration gradient exists. And a concentration gradient is when you have more concentration in one area and less concentration in another area. All right, so we can see, think of these little blue dots here as being our solute. And this white part here would be our water or our solvent. So we can clearly see that there are more of these blue dots in one area compared to the other area. So this forms a gradient. Now, this could exist just in, let's say, a glass of water, or it could occur across the cell membrane. So you can see that this area right here is our phospholipid bilayer. So sometimes a concentration gradient can exist between the intercellular fluid and the extracellular fluid. So this concentration gradient, once again, it means that there's more concentration in one area and less concentration in another area. These concentration gradients can be a driving force, which means that molecules or substances can be forced to move from one area to another area. Now, in some cases, these substances are going to move from areas of high concentration to low. And so that's what we're seeing in this picture here, where there is a high concentration of molecules inside, and there's a low concentration outside. And this is showing that the driving force here is that these little molecules or these little substances, whatever they have their own little vibrations, and these vibrations make them bounce off of each other. And so as they're bouncing off of each other, they're going to try to bounce away from each other. This is kind of like if I were to drop a whole bunch of bouncy balls in one area. The bouncy balls aren't going to stay in that area. They're going to start bouncing away from each other. And so as these molecules start to move from the high concentration area to the low concentration area, we say that they are moving down or with the concentration gradient. So it means that they're going from high to low. And you can see in this picture here that if we kind of pretend like this is our cell membrane, that this can happen in really either direction, but we're still going from the high concentration area to the low concentration area. So we're still going down or with the concentration gradient, whether we're going into the cell or against the cell. It all has to do with the concentration of the substances between the intracellular fluid and the extracellular fluid. Now, in some cases, like what we see with this image down here, substances can actually be transported from areas of low concentration to areas of high concentration. And when this happens, we say that these materials are being moved against or up the concentration gradient. Now, you can see here that we have a low concentration on the left side of the cell membrane and a higher concentration on the right side. So in this case, this substance or molecule, whatever it's called, is actually being transported from a low concentration area to a high concentration area. So it's going up or against the concentration gradient. Now, typically, when a substance is transported against its concentration gradient, there typically has to be ATP involved. And typically, when things are being transported with or down their concentration gradient, we typically do not need ATP. But there are some potential exceptions to that rule, so we'll talk a little bit more about these different types of transport and which types transport the materials with or down the concentration gradient and which ones transport it against or up the concentration gradient.